All right. Hello, everyone. Thanks for coming. Welcome to the first installment of this 2024-2025 season of the High Elevation 5-Needle White Pine Science and Management webinar series. This is our fourth year. We're super happy to start it. We have a really cool team that's going to be working with us this year, and we've also have a really fine selection of speakers from across the entire range of our beloved ecosystem that are going to be presenting their work for this year and half of the next one. I wanted to send greetings and a big shout out to all the members that we got to see each other in Baker City for the annual meeting. And I also wanted to encourage everybody to sign up for the next year meeting that will be in Bozeman, Montana. I will post some information about how to follow up with that in the chat. And I'm going to start with some introductions. Well, for the ones that we haven't had a chance to introduce ourselves in person or virtually, I'm Enzo Martelli, a PhD student in Karen Nelson's Restoration Ecology Lab, and I'm at University of Montana, Missoula. I'm co-organizing this webinar with two amazing other graduate students. The first one is Lou DeLuzzi, a PhD student in Daniel Ulrich Lab at Montana State University, and Karina Lee, a master's student from Diana Tombach's lab at University of Colorado, Denver. So if you miss any of our previous installments on either of the other three versions of the series, you can find the recordings or the talks on our YouTube channel using the link that I'm also going to paste on the chat box in a minute. So for today, it is my pleasure to introduce our speaker, Amanda de la Torre from Northern Arizona University. Amanda is an associate professor at the NAU School of Forestry, and she's interested in understanding how long-generation tree species respond to biotic and abiotic stresses using tools in genomic, transcriptomics, physiology, and epigenomics. Amanda received her PhD from University of British Columbia, Vancouver, and has also worked as a postdoctoral researcher at Umea University in Sweden and University of California at Davis. So if anyone has any questions for Amanda, please type them into the chat or wait and virtually raise your hand. And at the end of this talk, I can enable you to use your microphone. So with that, I'll turn it over to Amanda.
Hi, everyone. Thank you for the invitation to present at the webinar. Fortunately, my camera is not working, but thank you. Maybe later. So I would like to talk a little bit about some recent research projects in my lab about Whitebark Pine. And the title of the talk today is Using Genomics to Identify High Priority Areas for Conservation Across a Species Range of Whitebark Pine. Okay, well, we all know that climate change is, really bad. It's producing, temperatures to get warmer and heat and drought events to get more severe or more frequent. And all of this poses significant challenges to the development and survival of long-generation tree species and conifers in the Northern Hemisphere. In addition, this warming climate also brings more episodes of disease, and it can severely affect different species. So I would like to start my talk by giving a little bit of a background about the research that we do at the Forest Genomic Lab at the Northern Arizona University. We are located at the School of Forestry, and this is in Flagstaff, Arizona. We are focused on understanding the capacity of natural populations of tree species to adapt to new environmental conditions and diseases. And we aim to use this information to develop effective conservation and management strategies. So some of the research topics in our lab have focused on studying the genomic and evolutionary basis of phenotypic trait variation. We also have looked at plant stress response. This is response to disease and also to abiotic stress, like, for example, drought, heat, cold events. We also look at adaptation to changing environments by looking at genome-wide associations between genotypes and different environmental variables. We have generated a number of genomic transcriptomic tools like whole genomes or whole transcriptomes from different species, done a little bit of comparative genomics, and more recently, we have started to work in the genomics of endangered species, or also called conservation genomics, in species such as Whitebark Pine. Another project that we have is with mangrove species in Puerto Rico, looking at how to use genomics for restoration of riparian areas there. And last part is in relation to developing tools, as I said, genomics, transcriptomics, bioinformatics, epigenomics, physiology, ecology, evolution. So these are all the different fields of study that we look in our lab. So I like to mention that conifers are actually a very interesting group of plants, are very different to other plant species. If you work with pines, you probably know this already, but I would say that conifers have very long duration times. They grow really, really slowly. And something that it's interesting about conifers in general is that their morphology doesn't change much. You probably noticed that a lot of different pine species look very much alike in, the needles and the way they grow, compared to, like, for example, angiosperm trees, that they're so different because, well, of course, they have flowers and different leaf shapes and all of that. Conifers are not like that. And that is because they have very slow mutation rates. So we can see when we compare the genomes of different conifer species, we can find that they have not changed much, even, considering like dozens or hundreds of million years of evolution between the species. It's important to mention that is only in relation to the coding parts of the genomes. Another important characteristic is that they have huge genomes, and that had been a problem for all genetic studies in the past. It was very challenging to work with conifers because, well, first of all, the sequencing technologies were not ready to generate that amount of data, and then you need very strong computational facilities that are able to deal with this very large amount of data. So, but fortunately, in the last less than 10 years ago, we started having like genomic resources for all these conifers, and we can now use those as reference for genetic studies. And why this genome so huge? Well, the amount of genes is very similar to other plant species. The main difference with other plant species and other angiosperm trees, like for example, poplar or aspen or birch, is the amount of highly repetitive areas in the genome. And just, some years ago, nobody really knew what those non-coding parts of the genome were involved in. And well, we now know that they have an important function in gene regulation. And the study of this part is still in its infancy, and there's so much to do, but it's all very exciting. And these are some of the species that my lab has worked on, and the ones that have a star is the ones that my lab has collaborated to develop genomic resources of those species. So we can see that Whitebark Pine is at the top here. So let's talk about Whitebark Pine, because I know that's what's people here attending this webinar is most interested in, because you probably work with conservation or management of Whitebark Pine. Whitebark pine is an endangered species in the US and Canada. It has been severely affected by an exotic fungal called white pine bleach to rust, called with this caused by this fungal disease called carnassial revicula. Unfortunately, Whitebark pine natural populations do not have a major gene resistance to the disease. Like, for example, sugar pine or other fine needle pines have a major gene resistance. What does that mean? If the individuals have an allele for the resistance, then the tree would not die. That doesn't seem to be the case for Whitebark pine. However, we have observed quantitative resistance, but the rate that it occurs in natural population is very, very low. For this reason, many individuals or population Whitebark pine have been severely affected by this exotic disease. Also, population have been affected by mountain pine beetle and fire and drought, especially in Canada, but also in the US. So some of the recent projects in my lab have been to identify individuals or populations that are resistant to the disease, to white pine blister rust, and identifying what are the genes conferring resistance to white pine blister rust. Building a de novo transcriptome, which is a reference. And then we also participated as a collaborator in the genome project of Whitebark pine. And more recently, the research project that I'm mostly be talking in this webinar is the conservation genomic project in Whitebark pine, in which we collected individuals across a range of the species, including all national parks in Canada and most national parks in the US. So let's start talking about the white pine blister rust resistant experiment in Whitebark pine. So this was done in collaboration with Richard Nasco and Angelia Kelly from the Forest Service, and all experiments were performed at Dorina Genetic Resource Center in Oregon. So in 4 years ago, seeds from 128 parents were planted at Dorina, and 1 year later, young seedlings were inoculated with a strain of C. revicula, which produces a white pine blister rust. And since that time, infection symptoms were evaluated and are continuously evaluated until next year. So here is a photo, some of the photos showing the symptoms that this Whitebark pine seedlings after 18 to 20 months after inoculation of the fungus. So figures A to E show the different symptoms, cankers, and also the needles turning yellow, which are like very clear symptoms and are present in all fine needle pine species. And then figure F shows how non-inoculated family looks like, so they look healthy. So from all of these parents, we collected from a subset of these seedlings, we collected needle tissue for RNA sequencing 2 times. The first time was before inoculation, so time 0. And the second time was 4 days after inoculation. So from those needles, we flash frozen the tissue, take it, put it in liquid nitrogen, bring it from Oregon to my lab in Arizona, extract the RNA, and then send it for sequencing so we can do something called differential expression. And what we're looking at here is identifying which genes are conferring resistance to white pine blister rust. So we found a bunch of candidate loci, 599 differentially expressed transcripts. One of the main families were involved in flavonoid biosynthesis, and these are the chalcone synthetases. But we found genes that were differentially expressed that were part of different functions, very important biological processes, like, for example, secondary metabolite biosynthesis, response to water deprivation, response to UV light, and other like that. Some of them were upregulated, some others were downregulated, as we can see in this figure. In addition, with this RNA sequencing data, we created the novel neurotranscriptome that has 796 high-quality protein annotations. And we use this transcriptome to annotate the Whitebark Pine reference genome that was published earlier this year with David Neal as lead author at the University of California, Davis. And that was a collaboration among several institutions like Johns Hopkins University, University of Connecticut, and Northern Arizona University. All the differential expression and transcriptomic analysis was published also earlier this year and led by a postdoc in my group, Laura Figueroa, and it's available now if you want to read it. Okay, so let's talk about the conservation genetic study, because I'm thinking that the group attending the seminar are probably most interested in listening about some of the results. I have to say that this is the first time that I'm presenting the results of this conservation genomics project. So all the results that you're gonna hear today are still preliminary because we're working on the manuscript, but I'm excited to share with you for the very first time an update about this project. So for this project, we collected needle tissues from almost 800 individuals across the natural range of the species in Canada and the US. And that includes all national parks in Canada and most national parks in the US, in addition to other areas that were not part of national parks. So we received needles from collaborators, so we collected some of those. We extracted the DNA from needles, and then we used the DNA. We sequenced the DNA using exome capture probes that were designed from Candida loci for white pine blisterus resistance, and other important genes that we identified in previous studies, and also a random sample of coding and non-coding parts of the genome. So we, our final SNP datasets and SNP markers are just molecular marker that we use for genetic studies. Are 736,691 SNP markers that came from exons and 54,079 SNPs that came from intergenic regions. So why do we have two different SNP data sets? It's because for population genomics analysis, we would like to use markers that are potentially neutral, not under selection, so we can have a better estimation of population structure and genetic diversity. Excuse me. So the smaller data set will be the putatively neutral data set. Okay, so some of the initial results. Some of the initial results that we found. Well, something interesting that happened is that when we received samples from collaborators, there were some individuals where collaborators said they weren't sure whether they were limber pine or whitebark pine. And there were other individuals that were identified initially as whitebark pine, but turned out to be limber pine based on our genetic results. There was also a question, some people in Montana, they were wondering if these two species hybridized. Because, well, they are very morphologically similar, except when they produce cones. And they also have a sympatric distribution just part of the range. So if there were hybridization, there could be opportunities for crossing between the species. However, our study shows that these two species are very different genetically. If we see all of the big cluster here on the left side of the graph, these are all whitebark pine clustering all together very compactly. And on the left side, we have all the limber pine individuals, and they all come from two different locations, NAU National Forest and Lassen Volcanic. So in Lassen Volcanic National Park occurs something interesting. Some individuals are limber pine and other individuals are whitebark pine. However, it doesn't seem to be any hybrids between the two of them. So we conclude that these two species are very well differentiated. Okay, so let's focus on whitebark pine when we take limber pine out and we just focus on whitebark pine. And something that we see that caught my attention was that despite the very large natural distribution of the species, we see a high interconnection between the different populations. And what does that mean? It means that the populations, even though they're not so closely located to each other, and even though they occur at high elevation, they're still exchanging levels of gene flow, and this may be through seeds or pollen. So they are genetically connected, and that's why we're not seeing very strong clustering of populations. So we're concluding that there's extensive gene flow among populations. But there is a little bit of a latitudinal climb if we see that when we see that populations in Canada, and these are mostly national parks around the Rocky Mountains. So this is Jasper, Glacial, Kootenays here. So all this top part, the populations of Canadian parks across the Rocky Mountains are clustered here, and they seem to be very differentiated, or mostly differentiated from populations in California that are here at the bottom left of this graph. Okay, but overall, I mean, we see clusterings of individuals within populations, which is good, but overall, we don't see like big differentiation between regions, which is very interesting, I think, and also different to other conifer species I have studied in my group. Here are some ancestry plots. Here are some ancestry plots here. It's the same idea. We're looking at population structure here. Each line of this bar plot corresponds to one individual, and the labels below correspond to the number of populations. And what we see in here, when K equals 2, it means when there's just two clusters, we see in the populations in Canada and Canadian national parks around the Rocky Mountains are quite different than populations in this part of California, especially Sequoia National Park, Yosemite, in other parks that are located here. And when we included tree clusters, we see that there's more differentiation. This Canadian national parks here in purple, whereas the Californian ones are in red. So showing more differentiation between populations. Okay. So an important question about whitebark pine is, are we seeing signs? So this whitebark pine is an endangered species, right? So are we seeing signs of genetic erosion or, deleterious effects because, it's an endangered species, so population have been reduced drastically in numbers and in density. So we could think that we could see signatures at the genetic level that the species is actually struggling, right? But something interesting, our preliminary results show that genetic diversity is quite high in whitebark pine. In all populations, there's an excess of heterozygotes, meaning that the genetic diversity is high. And also, we are not seeing any populations that are showing strong signatures of inbreeding, which is very good. And I think from a conservation standpoint, this is a very important result because right now, we don't have any populations that were included in this study that is showing high or signatures of inbreeding, which will be, the consequence of inbreeding are very deleterious for natural populations, as you may know. I see that there's some questions in the chat, but I'm going to leave those for the end if you don't mind respond those at the end. Okay. So as I said, this is the first time that I'm showing results of this conservation genomics study. We're still working on this, but I think some of the main take-home messages for this work, and this again is all preliminary, are that, well, first of all, limber pine and whitebark pine are well-differentiated species. It is unlikely that they hybridize, unless, of course, there are some populations that were not included in this study that are showing hybridization, but from the ones that we study, we didn't find any signatures of that. We also found that despite the broad geographic distribution of the species, natural population seems to be lowly genetically differentiated, meaning that there's extensive levels of gene flow through pollen and seeds among natural populations. Also, there seems to be some level of genetic differentiation between populations in Canada and the US, especially populations in California that include Sequoia National Park, Yosemite, and Inyo National Forest that seem to be showing most of the differences in relation to other natural populations. As I mentioned before, genetic diversity is high and inbreeding is low. However, this does not mean that the species will continue to be resilient in the future if the threats continue, right? Disease, fire, and drought. We may see later on signs of detrimental effects. When we think about conservation, it looks like none of the populations seem to be entirely unique. And by that, I mean that is strongly differentiated from the rest, which suggests that all populations are important. I know not the message that conservationists want to hear. but, But it's this interconnection between populations, I think it's quite interesting. But of course, we can choose populations of high priority for conservation, taking into account other criteria. Like, for example, if we take into account the number of private alleles, so those are alleles that are present in just populations. For example, there's one population in Canada, Waterton Lakes National Park, that has a high number of private alleles, so it's important that that population will be conserved. Another thing that will be important, I think, to define high priority conservation areas is to look at populations that have a higher frequency of alleles that may show resistance to the disease, to white pine blister rust, or that high, or they may be like more resistance to drought and heat. So this is actually one of our next steps. We're currently working on that. So we'll be able to map those populations with higher allele frequencies and identify the exact geographic location where they come from. We are also working on genome-wide environmental associations and also machine learning methods, including gradient forests, to look at associations of genotypes and the environment. With that, I would like to finish my talk by acknowledging the people. This work wouldn't be possible, would not have been possible without the collaboration of many, many people that provided us with tissues from national parks in Canada and the US. Here are some of their names. And for that, my lab is very grateful to all these people who collaborated with this project. And we also have collaborators at National Park Service, Stephen Buckley, and Forest Service, the Bureau of Land Management, and also Parks Canada. This work was carried on by a postdoc group, two postdocs, Gustavo and Laura Figueroa, and some undergrads did all the DNA extraction. Funding came from the Bureau of Land Management in Montana and the National Park Service. With that, I conclude my presentation and I would like to respond to your questions now. Thank you so much.
Thank you very much, Amanda. It was an amazing presentation. We have a couple comments on our chat box and a couple questions as well. I would like to start by asking you, well, first of all, like I'm super impressed about with your results about like local differentiation of populations, and I'm really curious to know if you know whether it's possible to detect genetic erosion at a population level. And I'm asking this because I couldn't stop thinking about the different populations that were designated or proposed for the National Whitebark Pine Restoration Plan, and whether it's possible to know whether it's an ongoing genetic erosion in them.
So you were saying that if it's possible with genetic methods to identify genetic erosion, is that your question?
Yes, at a population level.
But yeah, of course it is. Yes. So for example, if we see that in endangered species that have very small populations, and this is, in different organisms, animals, plants, we see high levels of inbreeding. So inbreeding is when, two individuals that are closely related are mating with each other, producing offspring. And that is a signature that there are genetic issues there. So some red flags are very low genetic diversity in populations, or one population showing very low genetic diversity compared to the rest of the natural populations, high levels of inbreeding, and also presence of drift. We can also account for that. So all of that means that the populations or several populations are performed poorly, and they may have, you know, a lot of challenges surviving in the long term. So that, I think that's important to see in endangered species, because the main concern is that whether they would be able to survive changes in the climate, right? Because they are already, they already have like lower numbers of individuals, and they are suffering from all these different threats. Does that respond to your question, Ansel?
It does. It does. I actually it's really helpful to like listen to that. I was also like curious because what I was also curious whether it's possible to quantify this erosion at a local level af after a mountain pine beetle outbreak, say the populations in Yellowstone that have been severely affected, or here in Western Montana, like how much of that genetic pool has been affected by it just due to the mortality of it.
Mhm. Yes, yes, we can definitely look at levels of genetic diversity. If you have like a reference before the infection and after, and see where they find differences. It may not be seen right away unless there's like a bottleneck event, or like in a bottleneck you lose like, I don't know, 90% of the population, something like that. But if it's not like the differences in number of individuals is not so different, you may not see that right away. It may take some generations to show. So yes, but yes, definitely you can look at that, and that'll be very interesting.
Oh, okay, I'm gonna continue with one of our questions in the chat by Aaron Liston. Aaron says, "Great talk. How do you interpret the three samples that were intermediate between Whitebark and Limber on PC1 in your PCA plot? Did you also use a structure analysis of the full data set?"
Yes. Yeah, that's an interesting question. I haven't given a lot of thought of that, but I don't think those individuals are really hybrids. Um, I know that we we didn't include a lot of areas of symmetry in like sympatric areas in this study, and maybe that would be like a future study to look at those sympatric areas between limber pine and Whitebark pine, and see whether we find like the same results of contrasting results. We did run a structural analysis with the full data set as well, but we're not showing that in this analysis yet. I think those are, yes.
Thank you,
Mhm.
Amanda. There is another question here by John Van Gundy, and it says, "Is there any genetic study of whitebark pine populations isolated on sky islands in high altitude cirques?"
I'm not familiar with a study like that. No, but maybe.
Yeah. And I have a couple comments here from Andrew, Richard, Diana, that I will send you after the presentation here, because they're more directed to experiences that they have had, or recommendations on articles that address some of these issues.
Mhm. Great.
Let's see. Does anybody else has a question? Let's see. Well, if not, you can just let me know your questions and I will send them to Amanda. And with that, I want to thank you so much again for your time and your presentation. This was really, really interesting. I want to let everybody know that we're going to be posting this presentation in the coming days if you want to um, address it or revisit it again. And if you have, as I said, if you have any questions or comments, just send it to the webinar email that is posted on the Whitebark Pine Ecosystem Foundation webpage. And I'm also going to post it here on the chat for a minute. And with that, I also wanted to say that our next webinar will be on November 19th with Michael Murray, who's going to be presenting his talk, very disturbing agents of pressures on Canada whitebark pine. And with that, I want to thank everyone for attending, and I hope to see you all in a couple weeks. So thank you very much for your attendance, and we see each other in a month.
Okay, thank you so much.
Bye-bye.
Bye-bye.